I want to thank Samra, Perry, and David for the wonderful invitation to be part of this lecture series. And I am just so excited to be here to talk about spider silk. And I just love the theme of this lecture series, Mechanisms of Evolution, because it's an opportunity for me to grab all of you and to come just spend a few minutes in my world as I think of the mechanisms of evolution that shape extended phenotypes and repetitive DNA. So that's a little preview of what you're going to hear about. My presentation is divided into 4 parts. First, I'm going to give a background on spider silks. Probably most people don't realize sort of how long spider silk has been on Earth and how extremely diverse it is. Then I'll talk about what are the sort of the molecular underpinnings of spider silk, and that's this funny word here called spidroins. Then I'm going to talk about the molecular evolution of spidroins. And finally, I'll touch on the extraordinary mechanical properties that continue to inspire me to study more and more spiders. So spider silk is very ancient. So if you look at the fossil record, part of how you identify a fossil as a spider is the presence of spinnerets. And the purpose of spinnerets is to produce silk. So that means every spider that ever lived, whether it's extinct, it died last year, or it's alive today, has made or makes silk. So spider silk is really ancient. And if you look at sort of the age of our oldest spider fossil, spiders have been making silk for well over 300 million years, and that's part of why silk has just been really optimized so much over time. There's just been an enormous amount of evolutionary time for these silks to have diversified. And so there's 3 suborders of spiders. So we have our mesotheli here. So these are our segmented trapdoor type spiders. Megalomorph spiders, so these are tarantulas, trapdoor spiders, and their kin, and araneomorph, so these are true spiders, and these are spiders like jumping spiders, wolf spiders, and of course orb web weaving spiders. And a lot of people, when they think about spider silk and they think about spider webs, this is what comes to mind, sort of the wagon wheel shaped webs. But actually, the origination of spider silk was probably looked much more like this, where you have spiders that live in silk-lined burrows, where the silk is sort of fortifying the terrain, and they make little lids. So you can see this is a silken lid and with a little silken hinge. And here's a nice example of silken radial lines coming out of this burrow. So what this spider does is she sits in her burrow with a lid on top of her, and so she just holds those radiating lines. And the radiating lines are sort of a sensory system, so they expand her world. So spiders never evolved antennae, and so silk is a way that they can sort of expand their field. And so, and again, you probably think, wow, if that was one of the original purposes of silk, and if it's currently being used today, I wonder if silk transmit vibrations pretty well. And yeah, the answer is yes. It's just another one of its wonderful properties here. So spider silks are also extremely diverse. So, one of my missions in life is really to have people understand that spider silk is not one material, that we really should be talking about spider silks. So, and it's just so important that people understand that, because we have over 50,000 described species of spiders. And spiders are in nearly every terrestrial habitat, and then they even can be found in some aquatic habitats. So there's spiders that live in freshwater, and there's also some spiders that live in the intertidal areas. So they're everywhere. And every spider, whether it's a juvenile, a male, or a female, produces silk. So I've seen spiders that, pretty much from their second instar, so after they hatch, immediately after hatching, I don't see much evidence of silk use, but sort of after the next hatching, you start seeing silken lines. And spiders, unlike insects that the silk-producing insects, spiders use silk throughout their lives. So, from early stages of development all the way to being a mature individual and for reproduction. And most spiders produce multiple types of silk. That's what really sets spiders apart from other kinds of arthropods that make silk. It's how many kinds of silk one animal can make. So spiders rely on their silk for a whole variety of ecological purposes. And you can see that all of these really are related to fitness. So you could see, obviously, this is a spider holding that's her egg. It's an egg case, so she's deposited her eggs, and then she wrapped them up tightly in a bundle. And that silk then is protecting the eggs not only from things like, getting wet, drying out, but also a really, really big protection they provide is from insects that would like to parasitize an egg sac. Here's a spider, of course, catching food, wrapping prey there. This spider is using the safety drag line. So spiders are part of their bodies are sclerotized or hard, but their abdomens are soft. And so actually, if a spider were to fall and did not have a safety drag line, the falls are actually catastrophic. And so with their abdomens that they're not sclerotized, it means that it kind of tells you something about the ecology of spiders. So spiders are sort of feast and famine type predators, so they can go sometimes fairly long periods of time without eating. So when they do come across food, they have to really be able to eat a lot. And so that's part of the thinking as to why spiders have these, abdomens that can really grow and shrink just really rapidly. It's kind of like wearing, elastic pants to the Las Vegas buffet. You could just take advantage of that. Spiders, of course, also, they make shelters out of silk. So sometimes a shelter could be as simple as a leaf stitched together with a few threads to hide a little, or it could be a very, very thick silken funnel like this. And spiders, never evolved wings, but they are able to fly, and they're able to fly with silk. So this is an example of a spider dispersing. So they use a particular kind of silk, and they're able to sort of eject the silk, and just these lines are able to pick the animal up, and they start flying around. And spiders have been collected up to 2.5 miles up in the air, and they've also been collected 1,000 miles from the shore. So they are flying and dispersing long distances. Now, not all spiders can do this. So some spiders, especially things like megalomorph tarantula type spiders, they're not able to do this. And so their only way to get around is to actually walk. And so if you start thinking about, what kind of animals are more vulnerable to, human activity and also climate change, the spiders that can't disperse very well, they're very vulnerable. Basically, a new housing development could wipe out a species. So spiders can make, nearly all spiders can make many types of silk. And the silk comes out of this organ right here. It's the spinnerets. So spinnerets are thought to be developmentally related to limbs. And in fact, if you sort of watch a spider walk and watch their spinnerets, they are in sync with each other. So there's a neural patterning there that's common to it. And spinnerets, ancestrally, there were 4 pairs, so 8, it's a very spidery thing, the 8 legs, 8 eyes ancestrally. Primitively, they had 8 spinnerets, but no spider today has all 8. Sort of various combinations have been lost. And so if you look on a spinneret under higher magnification, you can see that there's little spigots, and it's these little nozzles that the silk comes out of. So if we kind of zoom in there, you can see these are the little spigots, and you can see the fibers coming out. And so, for me as an evolutionary biologist and a silk biologist, I think it's so cool that, this fundamentally important organ for spiders is also one of the most important sources of taxonomic information. So a lot of information about, how spiders are related to each other, we actually get them from spigot morphology. So different kinds of spider silks, and I'm going to tell you what the different types are, they're associated with a different type of spigot. And there's a lot of, for those who are interested in sort of, materials processing, there's a lot of structure function relationships to be studied there, like why are some spigots narrower than others, why are some wider. Not all spigots are necessarily circular. Some of them are sort of more like rectangular, like the crevice tool of your vacuum cleaner, attachment set. So that's one of my favorite spigot morphologies. And so every spider can make multiple silks. So here's one of my study animals. This is the silver garden spider, Argiope argentata. And this Argiope argentata on her web. So spiders that end up in my lab, what happens to many of them are I euthanize them and I collect just the abdomen. So here's the abdomen. The head was here, okay? The spinnerets here, the spinnerets are here. So I take the abdomen and then I peel the abdomen. So I peel the abdomen and just leave the spinnerets on. And it's kind of like peeling a grape. And I don't know if anybody in here has ever dissected a spider abdomen. It is truly one of the most wonderful things to do, because when you peel that abdomen, and again, it's like peeling a grape, spiders are so full of silk glands that the silk glands just start spilling out. And so so I took this abdomen and I peeled her like a grape, and I removed things like fat, the heart, stomach, ovaries. I removed what's not silk gland, and this is what you end up with. And yeah, it really looks like this amazing like sea creature. So you can already see that there's a lot of glands, and they're really packed in there. It's like compression packed inside the spider's abdomen. And the glands, there's many glands, and they're morphologically distinct from each other. And so here's the same image of these silk glands, and it's from studying them, I can show you sort of how I can relate particular glands to particular silks that are used in the spider's lifetime. So can you see here, there's really sort of brightish white glands right here. These are actually clusters of what's called aciniform silk glands, and they're tiny. There's actually hundreds of them there. And they're the silk that makes this white blaze in here. And it's also the silk the spider uses for the inner lining of her egg sac and also to wrap prey. So this kind of spider, when prey are in her web, they kind of look like white mummies in there. Here, this sort of yellowish gland here, this is the most well-studied spider silk gland in the world. It's the major ampulate silk gland. So from this silk gland, that's where we get framed threads. It also makes the radii of an orb web and the well-studied dragline silk. So that all comes from this silk gland here. And another one that's really visible here is these sort of greenish, it's so green it looks almost black here. These glands here that look like noodles, so not surprisingly, their names is tubuliform, so these tube-like glands. And these glands make this papery coating of egg sacs that make a very, very environmentally resistant coating that, the eggs inside can still get gas exchange, but they're protected from the environment. So and there's other kinds of glands in there, but these are just some easy ones that I could pick out for you. So super fun to dissect spiders and see their silk glands. So now I've showed you that there's a lot of kinds of silks. Spiders have been doing this for a long time. So let's now, dive in and talk about what is silk. So silk it's a protein. So once you wrap your head around that silk is a protein, then you realize, okay, genes encode proteins. And that was actually my motivation when I wanted to go do a postdoc at the University of Wyoming, because there was a lab at the University of Wyoming that published the very first little snippets of spider silk genes. And I remember reading that as a graduate student, and I was like, I just got to go there and, just cold called. Yeah. And so I thought that was a 2-year postdoc project. And I thought I would know everything about spider silk from that. And I was terribly wrong. But it set me on this path. So let's look at this spidroin set here. So spider silks are composed of spidroins. So spidroin is a contraction of spider fibroin. And it's a type of protein. It's a type of structural protein that's present only in spiders. Okay, only spiders have spidroins. And this is a cartoon of the silk glands of an orb web weaving spider. So these are those tubuliform glands that I had pointed out to you. In the other species, they were dark green, and that makes the outer silk of an egg sac. We have aggregate silk glands, flagelliform silk glands, piriform, aciniform, minor ampulate, and major ampulate silk glands. And so the glands are numerous inside an animal. So in this spider, there are hundreds of these aciniform glands, dozens of piriform, and the other glands occur in either just one pair or up to 3 pairs. And from each of these glands, what's inside of them is not like a spool of thread, but it's actually highly concentrated protein solution. So we call that silk dope. So it's an extremely high concentration. And so it's liquid in here, and it's actually in what's called a liquid crystalline stage. So it's partially assembled, but it can't be fully cross-linked, because spiders, if you fully cross-link, then the silk cannot flow through the gland, through the duct, and out through the spigot. So when you watch a spider spinning a web, or you watch, a spider on your windowsill, and you see that animal drop, what you're watching is actually near instantaneous production of an insoluble fiber out of liquid dope. And that is really an amazing transformation. We don't have any protein in our bodies that can go from liquid to solid that quickly. It's a very unique thing that spiders are able to do. So from these glands, starting with work done, I always think of it as like the, old school molecular biology, cDNA cloning methods, peptide analyses. From that kind of work, we've identified spidroins. So again, this unique family of proteins, we've identified spidroins that are highly expressed in each of these gland types. And webs are composites of different spidroins. So when you see a web, it's not just like, oh, that's the web spidroin. So for this web right here, 5 types of silk glands were needed to make this web. So this is why, when I think about silk evolution, it's not just about the silk gene. It's also about how these animals sort of mix and match and combine them to make these composites. And it's also about choreography. You actually have to have the correct moves to use silk in the correct way. So the 5 kinds of silks that were used in making this web is major ampulate silk. That's the radii, and a little bit of the frame. That is, most of it's like cropped out here, but it's the stiff silk that makes the frame. Minor ampulate silk is also called temporary spiral, and it's used during it's like a scaffolding silk. So it's like when the spider is constructing a web. So what happens during web construction is a spider has a frame, makes all the radii. It looks like she's making like a pie slice. Then she starts in the center, in the hub of the web, and she lays minor ampulate silk. And she starts in the center and she spirals out to the end. And minor ampulate silk is a dry silk. And once she reaches the outer edge of the web, she then uses that as a sort of a pattern, and then she lays down the actual capture spiral from the outside in. And the capture spiral is a combination here. So this is part of the capture spiral, and here's an enlargement of just a tiny segment here. There's a filament here from the flagelliform silk gland, and it's also covered with wet liquid glue, and that's the aggregate from the aggregate glue there. So again, like thinking about choreography, when that spider is laying down the capture spiral, she has to simultaneously draw 2 kinds of silks out to be laid down simultaneously. And that also tells you like something about probably maybe those spigots for those silk glands would be really close to each other on the spinneret, and yet they sure are. And I just got to give a little shout out for flagelliform. It'll always be my favorite spidroin, because it's the one that like I discovered. And it was like the first time anyone ever saw a flagelliform spidroin was like, was my clone cDNA. So it's always will be special. And then piriform silk, wherever you see silk crossing another silk fiber, or where you might have seen, imagine the edge of this web being stuck to a branch. Wherever you connect either silk to a substrate or silk to silk, you have to have piriform or attachment silk. And so there's at least 5 kinds of silk in this web. So again, like just realizing like spiders, they have to have all these silken tools, but then they're able to make something pretty darn cool. And so as a spider starts laying down their capture spiral, she starts from the outside, and she actually just paints herself into the center of the web, because that spider herself, if she walks on her capture spiral, she will damage it. So she carefully goes back to the center and then sits and waits. So what happened now is, trying to study these spidroins. I want to tell you about, a lot of work now has been done after the first few cDNAs and then the first few genes, and now we know a lot more about spidroins. So I want to tell you about the general architecture of spidroins. So what you're seeing right here is one major ampulate, or the dragline silk gland from a black widow. The scale bar here, that's 1 millimeter. So just give you an idea of how big or how small you might think of this major ampulate gland. This is the secretory tail. This is the area, the lumen, where the silk is stored. And this is the duct where the silk dope travels down and gets narrower, 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 and where the molecules align with each other. And so that's how you end up with a fiber coming out at this end here. And so all the spidroins that have been found correspond to this general architecture. One is they're all very large. So if you look at the coding regions, about the approximate protein weight, they're nearly all of them are greater than 3,000 amino acids. So we have coding region, just looking at coding region, a smallish spidroin is 10 kb of coding regions, and we have a spidroin from a black widow that's 133 kb of coding. That's absolutely enormous. And they're highly expressed, so we can really be, not surprisingly, they're really, really highly expressed in silk glands, and we really only find them expressed in silk glands. The transcripts, of course, are very long, and they're mostly composed of tandem repeats. So when you think of silk, you just got to get across in your mind that it's a study of functional repetitive DNA. So we have amino acid motifs here. It might be a string of polyalanine. It might be a glycine, proline, glycine type motif. So you see these motifs. These motifs reoccur over and over again, and you start seeing they occur in a pattern. And so they occur in these repetitive units, and these repetitive units are arrayed just like boxcars on a freight train. So that's how we get that. And then there's a non-repetitive amino terminal and a non-repetitive carboxy terminal regions. The amino terminal region is about 150 amino acids. The carboxy terminal region is about 100 amino acids. And those are actually the only two parts that really identify by sequence homology that these are all members of the same gene family. I'm going to show you that this repetitive region, sort of the boxcars on the train, they can evolve wildly different sequence, but sort of, the N and the C, the head and the tail of the train, they have more conserved sequence, and that has to do with the actual processing of the silk dope. That seems to be a common feature of all spidroins. And just one final comment here, a lot of the work I'm going to show you today was really, so much of it is really new since about 2019, 2020, and that's because with the availability of accurate long read sequencing. Most of my career, I worked with short read sequencing, so hundreds of base pairs. But when you're talking about repetitive sequence, you're not going to get that. It's very difficult to characterize very large transcripts with sequencing reads that can't span the whole length, because it's like the jigsaw puzzle with all the pieces that look exactly the same. It was very hard to assemble it. So now it's a lot easier to get data like this. So this is an example of a complete coding region of a spidroin. So this is one of the major ampulate spidroins from a black widow. This is the western black widow, Latrodectus hesperus. And I've shown here in blue, this is what this sort of hallmark of the sequence hallmark of the amino terminal region. And down here in blue, that's the carboxy terminal region. And sort of the meat in the middle, I've colored our glycine repeats, and I've colored our polyalanine repeats. And you can see, it's sort of very holiday themed here, but you can start seeing these repeat units. So you see these amino acid motifs that reoccur, but they reoccur in sort of sets. And so this is a repetitive unit here. And so different spidroin types have dramatically different repeat sequences. So here, these are two examples of two spidroins from the same species. So the one on the bottom here, this is a major ampulate spidroin, just like I showed you in the previous slide, but from a different species. And this is the repeat unit. This repeat unit is 34 amino acids. This is from the same species. This is an AK spidroin, a tubuliform spidroin, and this repeat unit is 180 amino acids. So basically, you're going to find this repeat unit tandem arrayed over and over and over and over again to fill up this very large giant proteins. Same thing, this 34 amino acid long repeat is going to be tandem arrayed over and over and over and over again to sort of fill the whole length of the spidroin. And I'm just going to color in serine, the amino acid serine, so S, and you can just see really obviously that this repeat unit is very rich in serine. This one has no serine, and obviously the length difference. And that also is all part of really trying to understand structure-function relationships here. So when you think about how these fibers, how these spidroins might interact with each other to make a fiber, if your repeat units are radically different in sequence and length, that has some implications for how fibers can get formed and what their properties would be. And as I'm a diversity biologist, and that was one of the really fun things. Like when I did my postdoc in a molecular biology lab, it was a lot more, in that lab, it was a lot more about, you want to learn a lot about one species, right? And so after I cloned the first flagelliform, the first thing I wanted to do was clone another one, right? And they're like, but you already have one, but it was sort of, but being a diversity biologist, it's like, but you won't really understand it until you start studying it in other species. So here's just some, this is some work I did before. So this is the Western black widow. We have whole genome sequence. We found about 25 spidroins in that black widow, and this is the depiction of the morphology of what the different silk glands look like. The scale bar here is 1 millimeter, so you can see the glands look totally, they're very distinct from each other within the spider. And then this one here, this is an ogre face spider. This is a nocturnal hunting specialist, and this spider makes this web here, and she holds it between her legs. In the spider literature, it's always described as a postage stamp-sized web. And the spider hangs upside down like this, holds the web, and when prey comes by, she stretches it out about, it can stretch to about 40 times its original size. Like she holds it, and then it gets enormous in area. And here are the glands. So these glands look kind of funny because this spider, during the day, looks like a twig. She's like a twig mimic. And so that means she has a very elongated, sort of skinny, cigar-shaped abdomen. So that has definitely shaped all her silk glands. So all her silk glands are very skinny and elongated, makes it very, very hard to dissect. But these are all the different kinds of silk glands in this spider. And this spider has about 13 spidroins. So it's always hard to predict. They looked about, morphologically, maybe as diverse as each other. But this type of spider has more kinds of spidroins than this type of spider here. So now you know what a spidroin is, and there's a lot of them and a little sense of what they look like. So how do they evolve? So I'm going to now tell you a little bit about a molecular evolution perspective of spidroins. And so the thing to keep in mind here is really start thinking about repetitive DNA, and what happens with all these layers of this hierarchy of repeats and what the implications are. So you've seen this already, and that from an evolutionary standpoint, I've told you that these are members of a gene family, but yet if you look at these repeat units that make up the majority of the spidroins, you can't actually sensibly align them to each other. That sort of concept that molecular evolution is positional homology, that falls apart when you look at spidroins. There is this repeat here, the first repeat, and the first repeat of a paralog, so another gene family member, there's no relationship there, right? So it's these repeat units are not evolving in a typical way, so that creates a challenge. But what we use instead to study gene family evolution is we use the non-repetitive amino terminal and carboxy terminal regions. So all our inferences about how the different spidroins are related to each other are largely based on these amino terminal and carboxy terminal regions. Now with whole genome sequence, for closely related spidroins, we can actually get some information from flanking regions, so we can sort of look at that. But even that is really only useful if the spidroins are fairly recently diverged from each other. If it goes too long, then flanking regions are sort of unalignable also. So if we align our amino terminal and carboxy terminal regions, and then you can make a gene tree. This is a gene tree we have for the different kinds of spidroins I've told you about. This is our flagelliform spidroin clade, our aggregate glue clade, piriform clade, egg case, aciniform clade, and major ampulate, the dragline, and minor ampulate clade here. And so, we can see things that can help us understand what happened is that, we know that major and minor ampulate glands, they look very similar to each other, and in fact, their spidroins do cluster together into, a major and a minor ampulate. We can see that, the two kinds of silks that are involved in the capture spiral, flagelliform and aggregate, they actually are sister groups on this gene tree. And that's just really interesting because actually having flagelliform silk gland and having an aggregate silk gland, that's actually one of a defining trait of being an orb web weaving spider, and other spiders don't always have these glands. So the glands occur together, and the spidroins seem to be most closely related to each other. So we do a lot of work in thinking about these evolutionary trees of our spidroins. But what about the spidroins themselves, kind of looking at that function there? So if you look at a single spidroin, so here I'm looking at the silver garden spider, Argyrope argentata, and I'm looking at the gene for prey wrapping silk. It's also that silk that made the white blaze in the web. So here it's coming out from this spider. She's wrapping up a cricket there. So the complete gene is about, the coding sequence is about 14,000 bases. It's made up of 20 repeats. The repeats are rather long, so it's 612 base pair repeats. Again, imagine a freight train, and these are the cars on this freight train. So, and you got 20 cars here. And if you do a crazy thing that people usually don't do when they study a single gene, is I start aligning parts of the gene to itself, I find that I have more than 98% nucleotide identity. So that means I took this repeat, this second repeat, third repeat, fourth repeat, and I basically aligned those repeats just to each other. So what does that look like? I'll show you just a portion of the alignment. So the way I made just to orient you here, the top line is the consensus of a whole bunch of these repeats, and the dot means that a particular repeat has a match to that consensus sequence. I'm showing you about a third of the alignment here, and can anyone see the one variable spot? Yeah, there's one right there. And it turns out to be a synonymous change. So in this part here, this is, so it's partly you can sort of see why, it wasn't until we really could get, long read sequencing that we could really start studying spidroins in a way in a much, much faster way. So to get data like this before, I was doing things like making large insert genomic libraries. I was doing a lot of physical mapping of clones in order to sort of convince myself that I really was reading repeat 6 versus repeat 7. It's really hard to know where you are when your sequences are this homogenized. And so you wonder, what kind of evolutionary mechanism is operating here? Are we seeing sort of, concerted evolution, homogenization happening of these sequences, or could it actually, the other hypothesis, one other hypothesis would be, is there actually selection for particular codons to be used in particular locations in a repeat unit? And I think that given how frequently I start seeing this kind of result, I think it's concerted evolution. It's a molecular process that's homogenizing these repeats to each other, whether you could have some gene conversion, you could have unequal crossing over, that is really keeping these repeats very, very, very near identical to each other. So I've now made a little tree out of these sequences, and so these are the the repeats from the garden spider. And you could see, and now I've combined, I aligned each of these repeats also with the repeats from some other closely related species. And you can see that all the repeats, they all group by species so that within a gene, you're homogenizing, right? But they're still similar enough across species that you can see that they're related to each other. You can actually align them. But it's a very odd kind of homology here because I'm aligning parts of the same gene to itself. So that's what we have on many kinds of spidroins. We see that kind of sort of homogenization of repeat sequence. And so this was enabled by long-read sequencing. And now doing even better than just sort of being able to read through a single gene, we can now also reconstruct in some of my best assemblies, we have it at the chromosome, we've reconstructed chromosomes, but other times we just have some very excellent long genomic regions, so some very long contigs. And here's what we've learned. So I'm just going to give you one example. So from garden spiders, so this is an orb web weaving spider, and I'm just giving you one kind of spidroin. So our genome sequencing, we've now done many species of Argiope, and they range from about 30, over little low 30s to upper 30s in number of spidroins, and about a third of those are major ampulate spidroin genes. So again, for some reason, there's been an expansion of major ampulate spidroin genes in these garden spiders. So the way to read this chart here is each row corresponds to a genomic region from one species. So this is the genomic region from one species, and we have, and all these major ampulate spidroin genes are on that genomic region, and in a separate genomic region, we have the other member of that cluster. So there is another major ampulate spidroin that we found, and it never occurs in the same genomic cluster as the others. And so the second row, this is from a second species, third row, a third species here. And the red arrow indicates the coding direction. So you can see here, that there does seem to be some sort of conservation, right? Like there just looks to be a lot of synteny here. So we have a lot of these major ampulate silk genes. So now let's look a little more closely at them. So this is that same diagram that was on the previous slide. And so we also did expression analyses. So we looked at the actual, the different silk lines, and we wanted to see where these major ampulate spidroin genes are expressed. And we know that all the ones that are in this genomic cluster, we know that they're all very expressed at a high level in major ampulate glands. That one that was off by itself, really interesting, it's actually not expressed in a major ampulate gland, even though its terminal regions closely allied with the major ampulate spidroins. It's actually expressed in the piriform or attachment silk gland. So there's been some sort of like duplication and co-option by another gland type. And for those of you who might be familiar with all the work on trying to make synthetic spider silk, starting off from the 1990s, when the DoD was putting a lot of money into, and other agencies were putting a lot of money into trying to make spider silk, all those models were based on making dragline silk out of two spidroins, MASP1 and MASP2. But clearly, maybe we kind of like, it's a lot more complicated than just MASP1 and MASP2. So, just trying to think about if you're trying to recreate the natural fibers, there's a lot of ingredients in there that was cryptic before. And we have evidence for gene duplication, gene loss, and gene conversion. Let me show you how I see that. So this is a gene tree made from the terminal regions of all the major ampulate spidroins in this genomic cluster from these three species. And so just kind of like quickly walking you through it, there's this MASP4, this group here where all the arrows are pointing to the left, that's right here. There's a very large MASP2.2 grouping here. This sort of yellowish MASP5 groups here. This sort of orangey MASP2.1, and this green MASP1, and the purple MASP3 is here. So you can see that what we're seeing is that like there were the ancestor of these species already must have had these higher level categories of major ampulate spidroins, but within a particular category, you can see, we can see that, get some insight into what's going on here, especially with gene conversion, gene duplications, and gene loss. So let's just kind of focus right here on this blue group here. So you can see here that our GIP trifasciata, the third row, has a MASP 2.2a, 2.2b, and 2.2c. And if you just look at the orientation of these three genes to each other, and you look at the direction of the arrows, and you compare it to the other species, it sure does look like MASP 2.2a. Probably all of those you would think they should have grouped with each other, but they didn't. In fact, what you see here is a grouping of these MASP 2.2s. It's by species here. So all our GIP trifasciatas, they all grouped together here. So there's the terminal regions are converting. They're looking a lot more like each other. So let's just look a little more closely at what's going on here with these three genes in this one species. So we're looking at these three genes, 2.2a, 2.2b, and 2.2c. So now I'm showing you the gene structure that now we've gotten from whole genome sequencing. So, and remember, all the early work was done with cDNAs, and so this was completely cryptic to us. So the line is an intron, and the blue rectangle is an exon. So we actually have a gene that has the N-terminus and the C-terminus. They're in their own exon. And then we have all these blue exons here. And if we look at, we're looking for these repeating units of sequence. So if we're going to look at the coding region here, and we find this repeat sequence that we had known about before from cDNA work, but now we know it in fine detail. And so we have this repeat unit, so of these three similar repeat units, that's sort of a lot of glycine, proline, glycine, the GPG, and a polyalanine here at the end. They're kind of similar to each other, but they occur in these triplets. So this repeat, the top repeat unit, the middle repeat unit, and the last one, those occur in a row. You can align them with each other, but you can see that they do vary. But then that entire unit then repeats again and again. And it repeats with 99.6 repeat identity at the nucleotide level. So this is kind of similar like what I was seeing with the aciniform spidroin, except in this case here, where there's an intron that's interrupting it, and that's actually where this line, this gap is right there. Here's MASP 2.2b, that's the one here. So MASP 2.2b also has a very similar repeat units grouped into an ensemble repeat interrupted by an intron. And again, this unit, including its intron, is arrayed over and over and over again, 17 times, 98%, about 98% repeat identity at the nucleotide level. And then you might think, oh, okay, so they always have a lot of these small exons, but that's not true because 2.2c is basically has one intron and a big exon, and it has this repeat unit, which looks quite similar to the other two in the group, and this occurs 26 times with a little over 97% repeat identity at the nucleotide level. So we're seeing this, these higher level repeat units, they're highly identical to each other. And I can now take this repeat unit, this triplet that I'm calling an ensemble repeat, I take this one and I can take this one and I can align them to each other. Again, a very weird thing to be doing. We're aligning these repeats from the same gene and across genes. And you can totally align them, and a pairwise amino acid identity of 86.5 to 98.5%. Like, some of these are so similar, like they almost look like alleles, but we know they're in different genomic locations. So these ensemble repeats also evolve that way. And so it looks to me, if you could see this, there clearly is stabilizing selection for keeping this glycine, proline, glycine type sequence with this polyalanine that's keeping that, there's selection that's maintaining that, because look how identical, like these sequences look the same. It's amazingly identity across a single gene and also across the different genes, but in the same cluster. And then now it's going to get a little weird. So now we're going to talk about these introns that have appeared. These introns are also repeating sequence. So this is, again, not something that people usually see, but if you look at the introns that are in between these repeated exon sequences, the green triangle means that there's excellent sequence identity there. And so I can easily align all the introns to each other. So I'm aligning introns from the same gene. And that's what the intron looks like. It's about 599 base pairs. It occurs 24 times in this gene. In MASP 2.2b, it has a different intron, but that intron is also repeated. It's a much bigger one, over 1,800 base pairs, and it repeats 17 times with really, and I'm going to show you with how near perfect fidelity. And so if we thought it was cool how the exon regions, that coding sequence, all look so similar to each other, even more amazing is that now these introns are equally high identity to each other. And here's the 2.2c that sort of, didn't want to have all the flash and dash of these repeated introns. It instead just has one intron, and this intron doesn't have sequence identity with these other ones here. So there's strong homogenization of introns within each gene. So this green one here, this green intron type that's in 2.2a that occurs 24 times, it's 98.8% identical, and the blue one is 97.9% identical. And actually, it's a smidge more higher identity than the exonic sequences were. So that's just, that's, again, just thinking about this hierarchy of repeats. And so but if you were to compare sort of the blue one with the green triangle, and also with, the single exon that's in 2.2c, there's extremely low similarity. Like, you wouldn't have any reason to think they were homologous to each other. So there, you have that sequence between them, yeah. And so we conclude here that it's very likely that we have concerted evolution of repeated exons and repeated introns within a gene, and that there's stabilizing selection of these protein repeat regions. And I'm just going to go quickly through this. So these are the 3 gene architectures you've seen. And I can talk more about this to you later. But if you start looking at those other 2 species I had, these are their gene architectures. So the blue circles, they exist in the other species, and you can still align them to each other, and they will cluster by species. But even a really surprising discovery was that in this other species, there were repeated introns, except there were 2 types. There was like an A and a B type, and they repeat over and over again with over 99% identity. So again, this is more repeat sequence. And our 3rd species, you start seeing this. So again, this kind of repeat sequence there. And now, why does it matter that we have these, what's being selected for? And you think about these mechanical properties. So a lot of people have heard about the spectacular mechanical properties of spider silk. And really, it has to do with that it's not just one material, that there's multiple materials. So you could find a spider silk that's great for whatever your application is. You want one that's super stretchy, there's one. You need a silk that works underwater, there's a silk. You want one that's super tough, there's a silk for that. And so the different kinds of silks perform different ecological purposes, and so not surprisingly, they have different mechanical properties. And there's work to show exactly what underlies the properties. At a coarse level, we can relate it to particular amino acid motifs. So glycine, proline, glycine is one of them. And so wherever you see the kink in this coil, that's the kink introduced by proline. And so this makes this super, super stretchy capture spiral that's really not very stiff and can just really extend to over twice its original length. By contrast, if you look at the frame and radii of a web, that kind of spidroin has a lot of what we call polyalanine, and polyalanine interlocks with each other, and it makes crystalline regions, kind of like Lego blocks here. And so you get a silk that is much stiffer and can't extend quite as far as flagelliform capture spiral silk. And so spider silk is wonderful, but spiders can't make enough silk for all of the uses people might have thought of for it. You might have heard bulletproof vests, airplane parts, bandages, drug delivery systems. So people take the lessons learned by looking at these evolutionary designs, and you can use recombinant technology and start gene editing and inserting genes into organisms that are a little easier to care for. So all spiders are predatory. That's sort of a challenge there. And so just to sum up here, I'm just going to share with you my rules of silk. And so silk has hierarchical genetic variation. I think of this hierarchy of repeats at all levels, amino acid motifs. I have these repetitive units. The repetitive units can be combined into ensemble repeats, which even includes introns. We have a diversification of spidroins. We have diversification of silk glands. And we have this fabulous over 50,000 species diversity. And if you have the dynamic evolution of structure-function relationships that you can see with looking at spidroins. So thinking about silk and spider webs, they're this remarkable extended phenotypes, thinking of all the different kinds of selective pressure on webs. We have multiple direct connections to fitness, silks that are used in reproductions, prey capture, protection. They have the different mechanical properties, all from just a single gene family. And we have optimized designs that can be used for biomimetics. And so there's a lot of spiders to work on. This is one of my favorite spiders I'm working on now. And so this is actually an orb web weaving spider. And her orb web has been reduced to this single ball. So this ball is where the capture spiral and that glue are. And she's reduced it to that. And so we're studying the spidroins combined with studying the behavior to understand how that worked. How did evolution go from an orb web to this? I mean, it seemed amazing just to make an orb web, but how do you do this? And I just want to thank all my collaborators and my funding sources, and I'm happy to take any questions.
And please use your microphones. Thank you.
Thank you. That was very nice. A natural question is there any relationship between silkworm silk and spider silk? Do they share any properties, or are they very far genetically removed?
That's a great question, and that's actually why I had called this rules of silk and not rules of spider silk, because now, as other labs around the world and also groups that entomologists I'm collaborating with, we're finding that what we've learned from spider silks does actually apply to insect silks. Now, insect silks are convergently evolved, so they're independently evolved compared to spider silks, but different insect groups have also independently evolved silk. So silk has evolved multiple times, and basically, okay, I'm a spider biologist, and so realize I'm a little bit biased here, but I don't think there's any other group of animals that can make silk as well as spiders can. But what we do find is that if there's a particular insect silk, you can find that kind of silk in a spider. So you mentioned silkworm silk. Silkworm silk, if you actually look at that amino acid sequence and you look at jumping spider major ampulate silk, if you put those two into NCBI BLAST, it comes first. It all gets confused, and it thinks your proteins are DNA because it's glycine and alanine rich. So it gets confused with that one. But it basically comes up with it's a score of 0. Like it thinks it's completely like they're identical. So it's an amazing case of convergence at the molecular level. The properties are similar but not quite exact, and so that's actually one thing I'm working on. It's like, what is it about the spinning system of a silkworm silk versus a jumping spider that, despite they have the same very similar recipes, the spider silk still seems to outperform the silkworm silk. But silkworms have a big advantage. They can make a lot more of it.
That was an incredible talk. Thank you so much for sharing with us. I'm curious about the behavior, like the diversity of behavior that's going on. And I think you mentioned earlier in the talk that the spiders can produce silk throughout different ages. And so I'm curious about if the behavior itself are there some learned behaviors, or are they just innate? Because it seems so complicated. So I'm just curious about that.
Yeah, that's a great question. And where I'm hoping sort of where our biological tools can take us, it's like a dream for me is to actually understand the genetic basis of silk spinning, and web spinning in particular, because it's clearly ingrained hardwired behavior. So if you look at orb web weaving spiders, basically, the young spiderlings almost never actually meet their mother. So they have a reproductive system like an annual plant where the mother lays an egg sac, and it's part of why there's so much investment in that silk. So she's putting proteins in the eggs, but she's also really wrapping them really tightly with silk to protect them from the environment. And so they come out and they sort of have to fend for themselves. And they just start, at some point, can start making little orb webs. And depending on species, it's so stereotyped. Like an arachnologist can actually walk through a forest and look at a web without resident spider and can really guess who made that web. So we know it's a stereotype behavior. There's one group of spiders that we think there is some learned behavior with what they do, and that's with a spider that's it's called an araneophage, so it eats other spiders, and it seems to sort of be able to interact with host spiders and sort of modify their behaviors based on what the host is. But that's a little unusual.
One last quick question.
How metabolically expensive is it to make all of this silk? I mean, because it's a lot of protein that the spiders have to sort of process.
So the proteins that are really, really abundant in spider silks are proteins that, if you look at sort of how expensive it is to make a protein, the bulk of spider silks are made from sort of the slightly cheaper amino acids, like a lot of glycine and alanine. That it's almost like many textbooks actually define silk as glycine and alanine, serine rich materials. Not all spider silks do differ by their amino acid composition, but they do seem to be sort of perhaps not that expensive. Spiders also recycle. Many of them do recycle their silk, and I think I think the cost of spider silk is also why silk is such a fantastic material, because it's protein. There's an animal invests in it. It can only make so much of it. And so it's been optimized to have these very amazing properties at an incredible light weight. So, for really the minimal amount of material.